Now we are going to study proteins. Proteins are derived from the word proteose, that means of the prime importance. The main sources of proteins are milk, cheese, pulses, peanuts, fish, meat, etc. and the responsible and the functions of the life. These are required for growth and maintenance of the body. Proteins are the polymers of the amino acids. Okay, so the amino acids or the amino acids, they are the bifunctional molecules, that means having the acidic carboxylic group and as well as basic amino group. So both amino and carboxylic acid are attached to the same carbon atom. Now let's classify the amino acids. The amino acids are, depending upon the relative number of amino and carboxylic groups, they are divided into three types, that is neutral, acidic, basic. As neutral, that is COOH group and NH2 group are same number, okay, like glycine, alanine, valine. In acidic, number of COOH, that means carboxylic group is greater than NH2 amino group, that is aspartic or glutamic acid, and in basic, that amino group is more than the carboxylic acid group, like lysine, arginine, histidine. Now, on the basis of synthesis, whether they are essential or non-essential. Essential means they can be synthesized in the body and are important in diet, and we take it from outside the body, like valine, arginine, isoleucine and lysine, leucine. Okay? And non-essential are which are synthesized by the body but they are non-essential, like glycine, alanine, glutamic acid, aspartic acid, etc. Now what are the properties? The amino acids are colorless, crystalline, water soluble and high melting solids. They behave like salts due to presence of the basic amino group and acidic carboxylic group. We know that acid plus base make the salt. Since the amino acids form salt with acids and bases, their chemical reaction is similar to primary amine and the carboxylic acid. Zwitterions. In the aqueous solution, the carboxylic group can lose a protein and amino group can accept a protein, giving rise to dipolar ion known as zwitterion. For zwitterion, you must remember the dipolar word. This is neutral but positive and negative charges. So you can see the diagram. NH2 and COOH, it loses its H and it gains its H. Okay. Then isoelectric point. The pH at which this dipolar ion exists as the neutral ion, that means positive and negative charge is equal and it does not migrate to any of the electrode, that is isoelectric point. So the amino acids have least solubility in water at the isoelectric point, which help in their separation. Now except glycine, because it have H as the R group, all amino acids which are natural occurring are optically active because they are chiral, and chiral means asymmetric. They exist in both D and L forms. The amino acids like alpha amino acids, like natural one, have the L configuration, and the L amino acids are represented by NH2 group on the left hand side. So if NH2 is at left hand side, that is L amino acids. Now what are the structure of proteins? So the proteins, what are proteins? They are polymer of amino acids, and proteins are joined together by peptide bonds. So what is peptide bond? It is an amide linkage between carboxylic and NH2 group. Okay. Next, what are the products formed by linking of amino acids by peptide linkage? When amino acids are joined by peptide linkage, the product formed is known as peptide. So peptides are further divided into di, tri, tetra, depending upon the number of amino acids combined. Oligopeptides containing anywhere between 2 to 10 amino acids or the structures with more than 10 amino acids are known as polypeptides. Means if there are 2 to 10 amino acids, there will be oligopeptides, and if the amino acid structures have more than 10 amino acids, that means it has polypeptide. It is known as polypeptide. A polypeptide, if has 100 amino acids and the molecular mass higher than 10,000 U, it is known as protein. Okay. Next we have is classification. Classification on the basis of molecular structure. Fibrous protein means protein polypeptide chains. The chains polypeptide are parallel to each other and are held by hydrogen and sulfide bonds, and these are insoluble in the water, like keratin present in hair, wool, silk, and myosin present in the muscles. So globular protein are the polypeptides which are in the form of coils and have 3D shape. They are soluble like insulin and albumin. Now on the basis of the structure, they are classified as primary structure means linear sequence. Secondary structure means it has two types of structure, alpha helix and beta pleated. Alpha helix means in the form of helical structure, okay, in the right hand screw model you can say, but in the beta plated it forms all the polypeptides have a maximum extension and led side by the intermolecular hydrogen bonding. Then we have is tertiary structure, means if we will fold the secondary structure 2 or 3 times, it will form tertiary structure. Fine, it has a hydrogen bonds, disulfide linkage, van der Waals, and electrostatic forces of attraction. Quaternary structure means again folding of the tertiary structure. Fine, that means 2 or more polypeptide chains as the subunits will join with each other. Okay. Then we have is denaturation of proteins. When a protein is having change in pH and the hydrogen bonds are disturbed, so due to that the globules unfold and the helix get uncoiled and the protein loses its biological activity, and it is known as denaturation of the protein. Denaturation causes change in secondary and tertiary, but not in primary. Okay, primary remains intact, like coagulation of egg white on boiling, curdling of milk, formation of cheese, and when an acid is added to milk. How? Let me explain you. When we do curdling of the milk, that means we make curd from the milk, what we do is we just take a lactic acid, right? We add the milk in it, so we change its pH, we change its temperature, so it denaturates, okay, denatures. And then Choco's point that proteins act as the amino acids and provide the defense against foreign organism, and if there is a change in the sequence of amino acids, serious diseases can be caused, and if glutamic acid is changed by valine in the hemoglobin, it can cause sickle cell anemia.